of the metabolism pathway. What is mean by metabolism pathway? Mean a sequence molecules is converted to in the presence of the enzymes. If any glycogen mean carbohydrate along with the or biomolecules is there, that in the presence of enzymes, that is the sequence of the molecule is going to conversion is there. So during that, the glycolysis, it is they are going to performing to convert a sequence molecules like glucose to glucose 6 phosphate to pyruvate molecules. So in this glycolysis is the embedment Meyer-Hoff Farner pathway also they are called because it is discovered by the Gustav Embeden. Okay, Otam Meyer, Prof. and Jacob Carloll Farner. These scientists, these four scientists is very interestingly working there. Then they have to identify the pathway. Okay, that's why the name is coming. Embeden is one of the scientists. Meyerhoff is one of the scientists. The Parnas is the one of the scientists. The three scientists is very uh, by chemistical. Basically, these scientists are chemistry peoples. They are work out and they have to sequence molecules conversion in the presence of enzymes as a biochemistry related or molecule evolution. The glycolysis is the metabolic pathway that converted the glucose into the two molecules of the pyruvate through a series of the reaction. Mean glucose is a six carbon containing molecule. The pyruvate is coming to three molecule containing pyruvate molecule is there. Okay, now this molecule is going to conversion. Um, a brief about the glucose. Three main steps is there in the glycolysis. The substrate levels phosphorylation is the first one. Mean any substrate having requirement of the phosphate, that phosphate mechanism. For example, glucose is there. If attached to the glucose 6 phosphate group, it will convert to glucose 6 phosphate. Again, one more molecule is sufficient to attach to the glucose molecule. That is 1,6-phosphate. And I mean the position of the carbon attached to the phosphate. That is the phosphorylation mechanism occur. The conversion of the glucose phosphate is the to fructose phosphate and the formation of the two molecules of the phosphate molecule. Like glucose to glucose 6 phosphate is a one molecule, glucose 1,6 phosphate to another molecule it will come. And EMP, EMP mean embedded made of par Parnas. This is the EMP pathway, also called by somebody scientist. It is involved as glycolysis, explained by the embed of the Parnas pathway, and it is the name after the biochemistry. This goes to embed in Autumn, Mayer of, and Jacob oral pattern was the describe the steps involved in the pathways. This in this glycolysis, third steps of the glycolysis is the phosphorylation of the fructose 6 phosphate. The catalyzed by the enzyme is phosphofructokinase. What is the steps of the in the glycolysis? How many steps is there? Mainly for the glycolysis, which is the first step in the all types of the cellular response. Uh, Respirations in the anaerobic condition does not require the oxygen. That means it is an anaerobic condition, does not require for the glycolysis pathway in the requirement of oxygen, not requirement. The oxygen is present, the pathway will be continue onto the Krebs cycle and oxidation phosphorylase pathways is there. Okay. This is the main concept of the oxygen, oxygen importance in the pathway. Then if it is oxygen is continuous, is available there, the Krebs cycle pathway is there. and oxidative phosphorylation pathways. However, the oxygen is not present in the glycolysis. Some organisms can undergo fermentation into continuously produce the ATPs. In most of the alcohol, like ethanol production in industrial purpose, this pathway they have to target to the enhance the content of the ethanol by using microorganisms. And I mean the microorganism having a specific nutrient you have to supply, and in the reactor you have to go add the substrate. Then automatically the bacteria is going to anaerobic conditions. It will convert it to the ethanol. Okay, suitable nutrients is there, automatically the growth of the more bacteria is there. More cell division is occur in the bacteria. Then more production of the biomass and along with the substrate also produced. For example, we are targeting the glycolysis. The end product is the ethanol. The glycolysis generally in the pyruvate is the end molecule. But industrial oriented, I'm target to the ethanol production. You have to follow what is the molecule is in the major molecules in the glycolysis. You have to find out, then you have to give to the microorganism that molecule. Then automatically it is the organism utilizing that nutrient, then going to break down the small molecules from a big molecule to small molecules, downstream process. So this is the, but that organism also required for the more energy also. This is the glycolysis pathway. You just observe here for understanding. Before I want to show a little uh, a brief structure of this one for understanding the glycolysis process, glycolysis pathway. This is the glucose molecule. In the presence of hexokinase enzyme, hexokinase enzyme, it will convert it to glucose 6-phosphate molecule. This enzyme is going to acting as a enhance the catalysis process only enzyme. How it is attached to phosphate? The phosphorus is coming from ATP, adenine triphosphate, then break into the ADP. The phosphate was single phosphate, it will eluted. The single molecule is coming to the attached to here glucose molecule. That is called glucose 6-phosphate. Again, you have another enzyme is involved there, phosphoglucose isomerase enzyme. Mean in these steps, another molecule is there. You have to see here. This is the glucose 6-phosphate to glucose 1,6-phosphate, but they are going to direct to fructose 6-phosphate. Mean here it is telling na, here this according to this is the in third step of the glycolysis is the phosphorylation of the fructose 6-phosphate to catalyzed by the enzyme phosphofructokinase enzyme. Then what happen automatically the whatever the substrate of the product it will come. The glucose is a substrate, enzyme is the hexokinase presence. It will convert it to by using utilization of ATP, then convert it to glucose 6-phosphate. Now this is the product. Again, enzyme is there, same thing, position. Then another enzyme is involved this product. This product is going to now substrate acting. Then phosphoglucose isomerase in the presence of the glucose 6-phosphate, it will convert it to fructose 6-phosphate. Then what happened? Again, another molecule is there, fructokinase enzyme in the presence of ATP. Again, it will convert to ADP. Then isomerization is occur. Then automatically it will go fructose 1,6-bisphosphate conversion. So what happened? This is the fructose 1,6-bisphosphate is a end product of the glucose 6-phosphate to okay. Next, fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. This is the glyceraldehyde. Next molecule is going to you see here glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. And sometime the glucose molecule during the fourth step, the fructose 1,6-bisphosphate is directly involved to the dehydroxy dehydroxybutyrate, also converted DHAP. Now again the isophosphate isomerase enzyme it will convert it to glyceraldehyde phosphate. This is the hydrogenase enzyme in the presence of ADP plus and two phosphate groups, it will convert it to NADPH. So what happened? The phosphate is going to remove and NADH is converted. Then the glyceraldehyde 3 phosphate, whatever this molecule glyceraldehyde. Now you only this one. Okay, 1,3 glyceraldehyde phosphate molecule is there, and it will go converted to two NADPs utilizing and NADH is conversion. Then automatically 1,6 bisphosphate is converted glyceraldehyde. Now again, now again, this is a glucose kinase exokinase enzyme. Then converted into glucose 6 phosphate and isomerase enzyme is there. It will convert it to fructose 6 phosphate. Again, phosphofructokinase enzyme. The phosphofructose 1,6 phosphate is converted and aldolase enzyme in the presence of aldolase enzyme. It will convert to glyceraldehyde 3 phosphate. The glyceraldehyde 3 phosphate dehydrogenase enzyme, and I mean removal of the hydrogen, it will convert to 1,3 bisphosphate molecule and converted into the one molecule of the 3-phosphoglycerate molecule. In this, ADP molecules is utilizing and converted to ATP. Why the ATP is converted here? This bisphosphate is there now. In this one phosphate group is attached to the ADP, adenine diphosphate. Then it will convert it to ATP. Now, during this one, it will convert it to the one position of the phosphate is going to outside. That is attached to the ADP. That ATP is going to attach to the ADP, then converted to ATP in the presence of phosphoglycerol kinase enzyme. Then converted to 3-phosphate glycerate. 3-phosphoglycerate phosphate molecule conversion. Then again, it is a reversible reaction. Then again, 3-phosphoglycerate molecule. If any enolase enzyme is there, it will convert it to phosphoenol pyruvate molecule. Then phosphoenol pyruvate molecule in the presence of pyruvate kinase enzyme. Again, ADP is converted to ATP molecules. Then again, pyruvate is a end of the product molecule. This is the another understanding. This is previous we discussed not glycerol phosphate and dehydrogenase enzyme there, and 1,3 bisphosphoglycerate is there, phosphoglycerokinase, and ADP is attached to the this first C1 containing phosphor attached to the ADP, and it is converted into two molecules of the ADP and 3-phosphoglycerate, and in the presence of phosphoglycerate mutase enzyme. The mutase enzyme mean it is change the structure. Mean a position of the third position mutation is there. The mutase enzyme. Then it will convert to the 2-phosphoglycerate, and enolase enzyme is there. It will remove the second position of the phosphate. The phosphoenol pyruvate molecule converted and pyruvate kinase enzyme in the presence of two NADP and two ATPs are converted pyruvate. This is the over understanding the glycolysis pathway. Glucose to glucose 6 phosphate in the presence of ATP to ADP conversion in the presence of exose kinase, glucose 6 phosphate converted there. Then again, the glucose isomerase enzyme is there. Isomerase mean resonance structures is there. In changes there, the position of the change automatically the fructose 6 phosphate is converted. Then and ATP again utilized there. Then again, what happened? It will convert it to in the presence of glucose fructose 6 phosphate to phosphofructokinase enzyme. It will convert to fructose 1,6 bisphosphate. Then again, aldolase enzyme is there. the glyceraldehyde phosphate 3 phosphate is converted, and
Sir, video has been paused.
now coming, this is the glycolysis pathway from glucose molecule to 6 to 3 pyruvate molecule in the presence of different enzymes. In the presence of different enzymes, this big size molecule, 6 carbon content to 3 molecule containing molecule conversion. This is the another same pathway, but there is a very detail of the enzymes is there, the detailed enzymes, but all mechanisms are irreversible molecule. But this is a very important concept in the industrial ethanol production, like alcohol production purpose. Very important. You see, just the in this bacteria, the pyruvate molecule is there, and acetate is conversion, and formate dehydrogenase enzyme and converted plus pyruvate. It will acetate molecule is coming to CO2 and a hydrogen molecule. And lactate also, lactate molecules, so pyruvate to lactate, lactate to pyruvate, lactate dehydrogenase enzyme presence, NADP to NAD plus conversion. And pyruvate dehydrogenase, decarboxylase enzyme, the pyruvate molecule is there, pyruvate phosphate molecule is there. And pyruvate decarboxylase enzyme is there, it will convert to acetyl aldehyde plus CO2 molecule. Again, the dehyde, it will presence of NADH converted to Na plus NAD plus conversion. The alcohol is in the presence of alcohol dehydrogenase enzyme is there. The ethanol production is there. If, for example, in industrial level, I want to produce a more alcohol by using a special organism. What should I do? I have to focus on which organism is going to very suitable for breaking of the glucose molecule to convert it to the pyruvate to ethanol production. I don't want to the middle steps. Direct to which enzyme is going to give the pyruvate molecule or middle steps is requirement. That is depending on our interest of the conversion. Okay. But every molecule is acting as secondary molecule between the glucose to pyruvate to ethanol production. Okay. This is the one type of glycolysis pathway. Now coming to the Krebs cycle. Krebs cycle is a present in the every cell that cause the oxygen to produce the energy. Krebs cycle. The Krebs cycle is a present in every cell that use of oxygen to produce the energy. This metabolic pathway is used as an anabolic cellular principle, but also in the presence of the catabolism also. What is mean by catabolism? What is mean by anabolism? You have to learn it. Okay. Then you have to understand the concept of the uh, tricarboxylic acid pathway or pentose pathway. And TCA is the main, the full form of the tricarboxylic acid cycle. Tricarboxylic acid per cycle, TCA definition, is a series of the enzyme catalyzed chemical reaction that form of the key part of the anaerobic, aerobic respiration in the cell. This cycle is also called as the Krebs cycle and the citric acid cycle also. Mean the Krebs cycle is same, TCS cycle is the same, and Krebs cycle, TCS cycle, and citric acid cycle. If any one name is called, the starting molecule is as a acetyl-CoA, the pathway. First molecule is called as a what? Acetyl-CoA. From acetyl-CoA, where it will come? Acetyl-CoA. You have to note the point, where it will come. The acetyl-CoA, you have to find out. And it is called as a citric acid cycle or Krebs cycle. The major energy producing the pathway and occur in the mitochondria. Mainly this process is occur in mitochondria cells. Foodstuff enter to the cycle as a acetyl-CoA enzyme. CoA enzyme, and are oxidized. Now you see the acetyl-CoA molecule is foodstuff enter to the cycle as a acetyl-CoA enzyme A or CoA and are the oxidized. The first molecule is acetyl-CoA. Acetyl-CoA in the presence of citrate synthase enzyme, it will convert it to citrate molecule. Citrate synthase enzyme, it will convert it to citrate molecule. Again, this molecule is in the presence of citrate molecule is a substrate and enzyme is aconitase enzyme. Then it will convert it to isocitrate molecule. This is the product. Now the product is acting as a substrate in the presence of isocitrate dehydrogen enzyme. Now what happened? Isocitrate to in the presence of isocitrate dehydrogen enzyme, what happened? Dehydrogen means removal of the hydrogen molecule, and along with the carbon dioxide is going to be liberated from the cell. So the end product is called, here substrate is the isocitrate, enzyme is the isocitrate dehydrogen enzyme. Then what happened? It will convert it to product is alpha ketoglutarate. Then what happened? NADH is a cofactor of the molecule to enhance the enzyme activity to convert to NADH molecule, and CO2 is going to be liberated, and product is a alpha ketoglutarate. Again, the product is acting as a substrate and enzyme is the alpha ketoglutarate dehydrogen enzyme. Same carbon dioxide is going to removing, and NADH plus is converted to NAD plus to NADH conversion. Then again it will convert to succinyl CoA enzyme. Succinyl CoA enzyme. హలో అక్క నేను వేరే క్లాస్లో ఉన్నాక అన్న వచ్చేసి బాబన్న వాళ్ళ ఇంటికి వెళ్ళి మళ్ళీ చెప్పండి 1 minute. వాళ్ళకు ఓకేలా. Okay, sorry for inconvenience. So what happened in the presence of substrate is acting alpha ketoglutarate and enzyme is alpha ketoglutarate dehydrogen enzyme, and it converted to product is succinyl CoA enzyme. Succinyl CoA is a product. Now again the product is going to acting as a substrate. Then in the presence of succinyl CoA synthase enzyme, the ADP is a cofactor. Then it will activate the enzyme. Then it will convert it to ATP, guanine triphosphate. Guanine diphosphate, GDP. Guanine triphosphate is a energy molecule. So what happened? Again, the product is succinyl CoA is a substrate. Enzyme is succinyl CoA synthase enzyme. Then it will come product is succinate. The substrate again succinate is going to acting as a substrate, and succinate dehydrogen enzyme will convert to FAD to FADH2 conversion. FADH2 molecule. FADH2 molecule. Now product is called fumarate. The succinate molecule is going to presence of succinate dehydrogen enzyme. It will convert to fumarate. Now fumarate is a product. Then the fumarate molecule is a product. In the presence of fumarate is acting as a substrate. Then again in the presence of furnace enzyme, it will presence of water. In the presence of water, it will convert to as a product is malate. Malate to as acting substrate. The malate dehydrogenase enzyme is there NAD plus to converted to NADH molecule. Then product is oxaloacetate molecule. Oxaloacetate molecule is of end product of the TCA cycle or citric acid cycle or Krebs cycle. So in this TCA cycle, starting molecule is called what? Acetyl CoA is a molecule and converted into the citrate. End molecule is called what? In the TCA cycle, oxaloacetate is the end molecule of the content. So totally, how many ATPs produced here? One ATP is going to produce. One ATP is going to produce, like acting as a GDP. GDP. The more energy source of the ATP, then second source of the guanine triphosphate energy source. If you see as a energy sources, then how many NADH molecules is present mean converted product formation mean here between isocitrate and alpha ketoglutarate between one molecule NADH and one alpha ketoglutarate to succinyl CoA. These are two molecule. And here malate to oxaloacetate NADH molecule. NAD plus to NADH molecule. Total 3 molecules is the involved here NAD NAD plus to NADH molecule, 3 molecules. Now, how many FADH molecule formation? In FAD, one FADH2 molecule is formation from succinate to fumarate. During this process, FAD is a fumarate adenine dihydrate. Then it will automatically going to, then how many carbon dioxide is going to liberated in the TCA cycle? 2 carbon dioxide molecules going to remove here. Okay, this is the overview of the glycolysis and Krebs cycle. Okay, exactly which place the in the mitochondria is going to the cycle is started mean acetyl-CoA. This is the over view of the cycle is also known as citric acid cycle, and the tricarboxylic acid cycle is the fundamental metabolism pathway that occur in the mitochondria and eukaryotic cells, and the cytoplasmic of the cytoplasmic cytoplasmic uh, cytoplasm fundamental metabolism pathway that occur in the mitochondria of the eukaryotic cells and cytoplasmic of the prokaryotic cells. It playing the central role in the anaerobic respiration system, processing that where the cells are obtaining energy from the glucose. The other organism molecule will breaking them down and capturing the release of the energy ATPs. What is the definition? Mainly definition, the Krebs cycle definition. The Krebs cycle definition of metabolic pathway that is oxidized acetyl-CoA to carbon molecule derived from various fuel sources such as the glucose, fatty acids, and amino acids. It extract high energy electron from the uh, the acetyl-CoA molecule that transfer them electron carriers such as the nicotinamide adenine adenonucleotide. This is the full form NAD. Full form is nicotinamide adenine dinucleotide. NAD full name is called what? NAD plus nicotinamide adenine dinucleotide, and the FAD full form is flavin adenine dinucleotide. Okay, this is the flavin adenine dinucleotide, which become a reduced to the NADH and FADH to conversion respectively. These electrons carrying then participated in the subsequent oxidation process. The phosphorylation and electron transport chain reaction leading to the production of the ATPs. This work is focused on several components and reactions of the citric acid cycle. His work focused on the fumaric acid, crucially elemented from the cycle, which is earned him a Nobel Prize in the physiological and medicines in 1937. Uh, Sergeant Gyro, yeah, discovered was based on the study of the Pigeon bird's muzzle, known for the enduring oxidative capturing event after being the breakdown in the latter mill and release the aqueous solution. This expect exceptionally characterized the made pigeon bird breast muzzle in the idea objective from the investigation oxidative reaction. This is the to during the pathway conversion, establishment of the pathway. These are very evidence of the uh, background of the molecule identification reactions. Okay, in this mainly glycolysis when the glucose molecule goes through a special process called as the partial oxidation turn into the two pyruvate molecule. This whole thing happen inside the cytoplasm of the cell. So formation of the acetyl-CoA. From the glycolysis, the pyruvate molecule is moving into okay, pyruvate uh, molecule is moving to the mitochondria, inner part of this called as the mitochondrial matrix. In their there they go through something called as the oxidation, decarboxylation, and become two molecules of the acetyl-CoA. This change is the made possible by the special enzyme called as the pyruvate dehydrogenase enzyme. The Krebs cycle is like the superhighway where the carbohydrates, proteins, and lipids get the fully oxidized. They turn into the CoA, acetyl-CoA enzyme A, or other molecules in the cycle. Acetyl-CoA molecule is produced during the this process enter the cycle. Glucose get the complete breakdown here. The acetyl-CoA combination three carbon. This is the story of electron transportation. The next class, you just see exactly where process. Mhm. Okay. Now you see this is the what is the Krebs cycle, where it is started the molecule, where how it is starting molecule is there, what is the end molecule in Krebs cycle, the cellular response. You see, this is cell in cell. There is so many organelles is there. Like in that organelles, we are Krebs cycle identified the pathway in the mitochondria region inner membrane. That is the first molecule is called as a acetyl-CoA to citrate to another molecule during the conversion of the this is the pathway understanding the uh, to content how many ATPs GTPs ATPs how it production there NADP NAD plus how it is converted to NADH molecule and FAD molecule to FADH2 molecule conversion. This is the understanding the picture of the cycle. Hello. హలో ఎవరు? హలో చెప్పండి ఎవరు? చెప్పండి. వేసాకండి. 1 minute. 1 minute. అట్లా ఫ్రెండ్కి పోతే ఇది వాటర్ కాల్స్ వస్తున్నాయి అంటే వాళ్ళకు కంటిన్యూగా. Okay, now sorry for small interference. The mitochondrial electron transportation system. This is the ATP molecule. This is the ribosome.
లేకపోతే కట్ చేసేసింది క్లాస్ అయిపోయింది.
Okay. Now this is the structure of the ATP. Adenine molecule. This is the benzene ring along the pentane molecule is there. The amine group and ketone group is there. This is a ribosome. This is the triphosphate is there. ATP triphosphate. Okay, this is the Krebs cycle. We discussed it here already. The pyruvate there now in the glycolysis pathway. The following Krebs cycle diagram following the Krebs cycle diagram in detail showing the different steps of the structures and intermediate through the cycle, the enzyme and coenzyme which is catalyzing step in the TCA cycle. Whatever the glycolysis pathway during the pathway, the pyruvate two molecules formation, the pyruvate again pyruvate dehydrogen enzyme is there, the removal of the carbon dioxide and then converted into acetyl-CoA. The acetyl-CoA molecule is entered to the TCA cycle or citric acid cycle or TCA cycle, Krebs cycle. Okay, now this is the sequence of the molecule conversion. Now large C6 molecule to 3 molecule conversion. Again, it will go, again increase the molecule structures and downstream and upstream process in the presence of enzymes. There is a particular names is there. They, for understanding the metabolism purpose, they are given different names. The acetyl-CoA coming from pyruvate. Where pyruvate is where the glycolysis molecule containing end product. Now, the end product in the pyruvate dehydrogen enzyme is there. It will convert to acetyl-CoA. What is the starting molecule of the TCA cycle? In the TCA cycle, first starting molecule is first starting molecule is pyruvate, acetyl-CoA. Hello, anybody is there?
Yes, sir.
It is visible for your slide.
Yes, sir.
Yes, sir. Yes, sir. Visible.
Slide is visible there.
Yes, sir.
Yes, sir. Yes, sir. Visible.
Okay. So, what happened? This is the end molecule of the pyruvate pathway that is called as a TCA cycle oriented. TCA is the auxiliary state and starting molecule acetyl-CoA. The acetyl-CoA coming from pyruvate glycolysis end product. The pyruvate dehydrogenase. This is the to understand the concepts of the there is so many enzymes is there, but for understanding, we are going to the main junctions in the pathway. What are the main molecules is there? How it is going to conversion the isomerism, glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-phosphate, and how it will form the glyceraldehyde and glyceraldehyde in the presence of aldolase enzymes, it will convert to 1,3-bis-phosphoglycerate and 3-phosphoglycerate, how it is present, which enzyme is metase enzyme is present, it will convert to 2-phosphoglycerate and enolase enzyme is there, it will convert to phosphoenol pyruvate the phosphoenol pyruvate converted to pyruvate kinase. Then end product is the glycolysis pyruvate. The TCA cycle, whatever the pyruvate molecule is there, that molecule is entered to the mitochondrial region from the inner member to outside. The Krebs cycle started. If pyruvate molecule is there, the pyruvate dehydrogenase enzyme is there. I show now this is the molecule pyruvate in the presence of pyruvate dehydrogenase enzyme and CO2 removing and acetyl-CoA is conversion. The acetyl-CoA is removal acetyl-CoA enzyme now substrate in the presence of citrate synthase enzyme. It will convert to citrate to um, isocitrate and acronate enzyme is there. Here in this slide I'm showing only few molecules only. Citrate to isocitrate to alpha ketoglutarate to succinyl-CoA to succinate to fumarate to malate. These are for understanding during the 10th class is there. If you study in 10th class, we are going to only few steps like citrate to succinyl-CoA to malate to oxalate, and this is the TCA cycle or Krebs cycle. If you come intermediate, we are going to give two more intermediate molecules. If you study degree level, we give you the very detailed. And when you coming PG, very detailedly, this is the very detailed structures of the molecule conversion in citric acid cycle or TCA cycle or Krebs cycle, whatever it, and acronate to isocitrate to alpha glutarate to succinate to fumarate to malate to oxaloacetate. Your TCS end product is oxaloacetate. First molecule is acetyl-CoA. This is the overview class of this day. Next, we will meet tomorrow. Okay, this is over. Class is over.
Thank you, sir.
I will send the PPT also.
Okay, sir.
Okay, sir.
Thank you, sir.
Right.
If possible, please share this PPT in the group, sir. Mm.